Hello everyone, welcome to part 6 of the lecture. So in this lecture, we are mainly going to talk about gene expression and the sequencing techniques associated with it. So let's dive in this one. So this is going to be a bit tricky because I will talk about a few techniques, so bear with me. So the first thing is this particular image. We saw it in the previous tutorial where I talked about how DNA to RNA to protein is formed. So if you're familiar with this image, then I hope that you're familiar with this image. If you are not, then go back to the previous tutorial first and then come back. So here, let me talk about gene expression. So you can think of these as some sort of gene where you have like many exons in it. And through transcription, you have this primary RNA transcript. And through alternative splicing, we can have like multiple different types of mRNAs, which ultimately translate down to protein. So this is old story. We have talked about this in the previous tutorial. Now, if you have a higher gene expression, what it means is you have more messenger RNA derived from that. And that means you have potentially more protein sequence synthesized from it. Okay, that's the implication. So, if we want to measure gene expression, if we want to measure how active a gene is in a particular person or sample, then it is equivalent to measuring the amount of mRNA created from that gene region. So, if you think that this gene region is highly expressed, then that means a lot of messenger RNA will be created from it. And if we can measure the amount of mRNA created from each gene region, we can indirectly measure gene expression. That is the basis for RNA-seq. And RNA-seq will be to the rescue. So we will shortly discuss about RNA-seq, but let's discuss about what are the factors which actually influence gene expression. So first of all, this picture, we are all familiar with this. In lecture 1, in part 1 of this series, I talked about this. Now, we know that gene expression is mainly influenced by the tightness of the DNA wrapped around these histone proteins, right? That is the main contributor. So that is one thing. So if some portion of the DNA is loosely bound or is open and a gene is actually situated right there, then the gene is likely to be highly expressed. Okay. And if it is tightly bound, then the gene is likely to be lowly expressed or suppressed, basically. Okay. The other thing is, this is not another thing. The name of this phenomena is called chromatin accessibility. So if some place is accessible, that means it is loosely bound or it is open. So it is more accessible basically. Basically, this phenomena is called chromatin accessibility. And you will find this term in many parts of different research papers. So this accessibility can also be influenced by epigenetic markers like methylation, acetylation, etc., which we talked about in part 4 of this series. So basically, epigenetic marker is also influencing indirectly gene expression. And finally, there is a group of protein, class of protein, which is known as DNA binding protein. So if these proteins bind to DNA in a place which is near the promoter region, then also it can positively or negatively influence gene expression. So now let's talk about RNA-seq, which is the way we measure gene expression. So first of all, we have to isolate the messenger RNA from the bulk of RNA. And this is done in many ways, like technique and many other techniques are there, magnetic beads and other things. After you isolate these RNAs, mRNAs, you sort of use a sequencing machine or something of that sort to chop those into small fragments. So that is the next step. The next step is very interesting. We know that the process of creating RNA from DNA is called transcription, right? But there is a process called reverse transcription, which is done using some enzyme called reverse transcriptase. If you use this, then these small RNA fragments will convert to complementary DNA strands, which is also called cDNA. Now, why is it called complementary DNA? Look at this picture for a second. So you can see, just suppose for an example, this is your RNA sequence. This is particular RNA fragment is this sequence. So what is the complementary sequence of it? The complementary sequence of A is complementary base is T. Now, let me come to you a little bit later on. Let me go through this first. So complementary of C is G, G is C, A is T, G is C. So this is we all know. We all know this from lecture 1. Very basic thing. Now, why do we have A in place of U? It is because you know that in RNA, we don't have the T, right? We don't have any thymine. The T is replaced by U, which is uracil. And the complementary base of T is what? It is A, right? So similarly, the complementary base of U will be A. So this complementary DNA strand will be created by the reverse transcriptase for each case. That is why it is known as cDNA. But these are single-stranded DNA and not very stable. So obviously we are going to use our very famous DNA polymerase enzyme. We talked about it in the previous lecture as well when we talked about DNA replication. So DNA polymerase enzyme will create the complementary strand of these DNA strands. It will make the very stable double-stranded cDNA. Now understand that the reason we are actually not sequencing on these RNA fragments and we want to sequence on DNA fragments is because DNA double-stranded fragments are much more stable and it is much easier to sequence. So after this, we have these things and we know what to do. We will obviously have to amplify this using PCR. We have talked about this many times, and then we will do sequencing. So after sequencing, we are going to have many reads, right? We are going to have many small reads from this DNA, and we will have to do what? We will have to map this DNA, map these reads to the genes. So if we have now successfully mapped all those reads to all of our genes, we now successfully know how many reads are actually coming from each gene, which is indirectly a measure of the gene expression. Okay, so that is RNA-seq for you. Now, we can use different measures, we can use different scoring mechanisms to actually calculate the gene expression. Two very famous ones are FPKM and TPM. So I will talk about FPKM today. TPM is very similar. You can look into that. So FPKM has actually a very serious name, by the way. Fragments per kilobase of transcript per million mapped reads. Very, what should I say, hard name to pronounce, I should say. So let me describe it gradually. First of all, in this score, let's talk about measuring the FPKM value for a single gene. So suppose we have a single gene of a sample and we want to get the FPKM value for this gene. So first thing to do is we have to calculate the number of fragments mapped to this gene. Okay, that is the first thing. Now, the thing is the total number of fragments in a sample actually depends on the sequencing depth or coverage. So you know about sequencing depth from lecture 2, right? So if a particular sample is maybe 5x whole genome sequenced, then it will have 5x more reads compared to a sample which is 1x whole genome sequenced, right? So we have to normalize by total sample fragments. And also, if a gene is very long, right? Because genes can have different length. It can have 1 kilobase length or 10 kilobase length. So if a gene is very long, then obviously there will be more fragments coming from that, right? So we have to normalize for that as well, right? So that is what we are doing here. We are actually dividing by the gene length. So why do we have 10 cube here? We are talking about per kilobase, right? We are talking about per kilobase, that is why it is 10 cube. And we are talking about transcript per million. We want to take it to million scale. So we are giving 10 to the power 6 right here. Okay, let's now see an example. But before that, I want to just remind you of one thing. We are actually looking at fragment long here, not read long. We know the difference, right? We know the difference from lecture 2, the sequencing lecture. So basically, we have a forward read right here. We have a reverse read right here. And in paired end sequencing, these two reads together sort of mark the fragment, right? So this black thing is the fragment. And we know why paired end sequencing or paired ends and sequenced like fragments are more accurately mapped. We know that from the previous lecture. So that is why here we are using fragment now. And it is also for legacy reason, like people have been using it for a long time. So paired end sequencing is very common. So people haven't abolished this practice because it's a good practice, by the way. So this is an example I took from ChatGPT basically. I haven't created this myself. So we have 100 fragments, suppose mapped to a gene of interest, say gene A, and the total gene length is 1000 base pair. So 100 is here, fragment now mapped to gene. Gene length is 1000, so we are going to put 1000 right here, so 1000. Then the total number of fragments of that sample is 50 million. We have given 50 million right here, right? And 10 to the power 9 is from this place. So if you multiply this two, it's 10 to the power 9. If you do the math, then the FPKM value will be 2. Actually, any FPKM value which is above 10 is a very high FPKM value, very highly expressed gene. And FPKM value below 1 is generally lowly expressed. If it is less than 0.1, it is very lowly expressed. That's the thing. Now that we know this much, let's talk about something else. Let's talk about ATAC-seq. So full form of ATAC-seq is assay for transposes accessible chromatin using sequencing. First, I will talk about why I'm interested in ATAC-seq or we are interested in ATAC-seq. So we have already seen in this slide that the tightness of DNA or the chromatin accessibility sort of determines gene expression, right? So we want to actually measure. We want to have a measure of how accessible the chromatin regions are. This is something we want to measure throughout the whole genome. So how do we do that? We do that using ATAC-seq. Okay. So let me talk about ATAC-seq now. So in the ATAC-seq, let me break this down first. So you can see this picture right here. So this is basically, the nice histone structure which we are seeing for a long time. So you can see that some of the places right here are actually completely open, and obviously there will be some place here which are loosely bound, which have some like loose places. So now TN5 is basically a transposase enzyme which has a particular property. It places itself inside a DNA portion. It cuts that DNA. It simply cuts that DNA and inserts or pastes another piece of DNA into the cut site. So suppose we have released this TN5 with this red and blue sequence loaded. So it will find some region in our genome. It will place itself there, cut that genome, and place this red and blue sequence onto that place. Okay, that is its task. Now, TN5 cannot work in all places. So if you have some open or loosely bound chromatin region, it will be more accessible by TN5. That is the thing. That is the main property here. So the thing is, we preload this TN5 with some sequencing adapters. So what is sequencing adapters? So sequencing adapters are actually some particular sequences which are very much prone or which are very facilitating for the PCR amplification. So suppose this place has no histone wrapping, and so it's an open region, completely open. So many TN5 enzymes will be placed here and many adapters will be linked here. So this place will be a lot more amplified by PCR compared to these places which are tightly bound by histone protein. So you can imagine that we are going to have a lot more reads from these open regions during sequencing compared to closed or tight regions, because these open regions have been amplified a lot by PCR amplification. And that can be used to measure the chromatin accessibility. So it is very, very simple. And obviously you can imagine that we can use something very similar to FPKM as a scoring metric, because we can take different regions of the genome, we can map the number of reads in those places, and we can get the FPKM score. Now, legacy wise for ATAC-seq, we use RPKM, which is same as FPKM, same thing, but only instead of fragment, we just take reads. So we don't care about paired end. We simply care about how many reads are actually being mapped to these regions. So that's basically ATAC-seq for you. So basically now you know a lot about sequencing. You know about whole genome sequencing, targeted sequencing, ATAC-seq, RNA-seq, and what else? I don't know. Probably I have covered maybe one more in the previous tutorial. So 4 to 5 sequence techniques you already know. And that's basically all you need to know about sequencing if you want to dive into bioinformatics. Maybe a few more out there here and there. Oh, we also talked about bisulfite sequencing in one of the previous tutorials. So you know a lot about sequencing and other things and implications, and you are pretty much ready actually. But still, there are a few more things left. For example, we haven't really talked about protein structure, which is actually a very fundamental thing in bioinformatics. I also want to talk about a bit about cell-free DNA. So I will talk about those in the next lectures. So the link will be in the description of the next lecture. Thank you everyone. So hopefully I will see you in the next lecture.